Hey guys, today's lesson is going to be about cancer, cloning, and stem cells, which might seem like a weird combination of topics, but they all have to do with cells. So we're going to be talking about the relationship between cancer and cell division, as well as how cancerous cells form, spread, and just behave in general. Then we'll briefly touch on two different treatments for cancer. Then we're going to get into the process of cloning, and then as also looking at the pros and cons of cloning. And then lastly, we're going to be looking at stem cells, what they are, different stem cell treatments, as well as the pros and cons of stem cell research. So first of all, the cell clock is just a biological clock that regulates how often a cell divides. So when a cell gets to a certain age, it usually stops dividing. At that point, it's just not as healthy, and it's better off to just kind of let that cell die off and produce a new cell by a younger, healthier cell dividing instead. Um, when a cell specializes, it usually stops undergoing mitosis and cell division as well. The more specialized a cell is, the less often it will divide. So skin cells, which aren't very specialized, they look very close to just like a normal, kind of ovalish cell that we would learn about in like grade 10. Those tend to divide more. Muscle cells divide less. They're kind of like long and stretched out, so they look a little bit weird. They're more specialized. They don't divide as much. And then nerve cells are very highly specialized. A neuron looks very different from the like oval cell that we learn about, and they really don't divide at all. Spermatogonia, which are these sperm-producing cells inside of the seminiferous tubules, they're one of the few types of cells that will divide an unlimited amount of time. But cancerous tumors will also divide an unlimited amount of times. Their division isn't regulated at all. They just divide uncontrollably and at a really, really accelerated rate compared to any other cell that you would find in the body. So cancer refers to a very broad group of diseases associated with the uncontrolled or unregulated division of cells. That's why it's so hard to like cure cancer, because cancer isn't just one disease, it's like 100 diseases. Both cell life and cell division is supposed to be regulated by genes, but in cancerous cells, those genes become mutated or fail some way to regulate the cells. And there's two main types of genes that are affected by these mutations in cancerous cells. There's proto-oncogenes, which are the genes that promote cell growth. So if proto-oncogenes aren't turned off, then the cell just keeps growing and dividing and growing and dividing and growing and dividing, and it never slows down and never stops. In a healthy cell, those proto-oncogenes would be turned on during the division portion of the cell's life, but then would be turned off during the interphase portion, which should make up about 90% of a cell's life. So yeah, those proto-oncogenes are only supposed to be on about 10% of the time, will be on 100% of the time in cancerous cells. And then the other type of gene is called a tumor suppressor gene, which regulates and promotes cell death, which is called apoptosis. So in cancerous cells, those tumor suppressor genes are turned off. So instead of a cell dying when it's supposed to, it just keeps on living and dividing and growing. So the continual replication and division of these cells results in an abnormal cell mass, which we call a tumor. Cancerous cells also don't specialize into specific cell types as they mature. So, when a skin cell divides, those two new daughter cells will specialize into skin cells. When a cancerous cell divides, it doesn't specialize. It also doesn't carry out any normal cell function, so it's not like contributing to your body in any way. They also don't adhere or stick to each other very well, which allows parts of the tumor or cells off of the tumor to separate and then spread throughout the body by traveling through either your blood or your lymph. Benign tumors are usually made up of cells that divide, but they don't tend to spread or invade other tissue. Malignant tumors will spread through to other parts of your body, usually by like breaking off and then traveling through your blood or your lymph. That process of traveling somewhere else in your body is called metastasis. So you may have heard before that someone's cancer like metastasized. That means it spread somewhere else. Cancer treatment usually involves either radiation or chemotherapy or both. Radiation therapy is used to damage the DNA of cancerous cells. And if you damage the DNA enough, it will prevent them from reproducing. It'll stop the tumor from growing. And then the body can eliminate the cancerous cells as they die, the same way it would eliminate any dead cell matter. Chemotherapy is using drugs that will target and interfere with any cells that are dividing. Since cancerous cells are always dividing, the chemotherapy drugs will be able to kill off the cancerous cells. The downside is, of course, that some of your other cells will also be dividing. So that's why chemotherapy can be really damaging to the body, because it doesn't only kill cancerous cells, it kills any dividing cell. And that will include some of your body's normal cells, especially cells that divide more frequently will be targeted more by the chemotherapy. Radiation, by the way, also damaging to your normal cell DNA. The same way any kind of radiation is going to be damaging to your body. Okay, so that's it for cancer. Let's move on to a cooler topic, cloning. So all somatic cells in your body should have identical DNA, since the DNA is copied during the S part of interphase. And then when your cells divide through mitosis, both of those new daughter cells now have identical copies of the original DNA. When you clone something, you're going to use the DNA from one of the somatic cells to make an identical version of the original. So if I wanted to clone myself, I could take the DNA from any one of my somatic cells and use that to clone me, if that was legal, which it's not. So the clone would have the exact same genetic makeup as the original. However, clones will not act or behave and might not even be the same looking as the original, because a lot of physical as well as behavioral traits are affected by environmental factors. And the environmental factors that the original organism grew up with will not be the same as the environmental factors that the clone grew up experiencing. So when you think of things like height, for example, or weight, both of those are affected by environmental factors, not just genes. And so the clone might be a very different height or weight compared to the original. And again, behavioral factors very much influenced by the environment. Okay, so these are the steps of how to clone something. So first, you take the nucleus from any one of the somatic cells of the animal you want to clone. Then you're going to take an unfertilized egg from any organism really, but preferably one that is similar to the one you're cloning, and remove its nucleus. That process is called enucleation, the removing of the nucleus. Then you're going to take the nucleus from the somatic cell of the thing you want to clone and put it into the enucleated egg. Then you will allow that egg to just develop and divide for a few days, and then you will implant that developing cell mass into the animal. Again, similar to the thing that you're trying to clone. Usually it's the animal that you got the egg from, and then you just wait for the clone to be born. So this is what that looks like in pig form and sheep form. So for instance, let's look at this sheep A and B here. So we're going to take a body cell or a somatic cell from sheep A. That means that it is sheep A that we are cloning. We're going to take out the DNA, or take out the entire nucleus, which is where the DNA is. Then you're going to go to sheep B. We are not cloning sheep B, but we are going to steal its egg. You're then going to denucleate it. So you'll remove the nucleus. So there is now no DNA in sheep B's egg. We then put in the DNA or the nucleus from sheep A into the egg from sheep B, and then you just kind of let that form like a cell mass, and then you implant it into, in this case, sheep C, though you could have also implanted it into the uterus of sheep B, and wait for the clone to come out. And again, the clone is going to be a clone of whichever organism we took the DNA from. Okay. Yeah, all of these are kind of just showing the same sort of process. You can look through them, decide which one makes most sense to you. The upsides of cloning is that in the future, they're hoping to use it for organ replacement. So as you may know, there's like a major organ shortage in a lot of countries, including Canada, meaning that most people that need an organ transplant aren't going to get it. They're going to they're going to die before an organ becomes available to them. In the future, one of the things that they're looking at to reduce organ transplant list wait times is to take cells from the person who needs a new organ and essentially like clone an organ for them, grow an organ out of their new cells. So yeah, cloning could be used to make a functioning organ from a few cells of the original. It's also cloning, that is, also useful to scientific research. In the past, twins have been used a lot with scientific research around genetics, but twins aren't always easy to come by. And also, like, there's a lot of history around the use of twins in like Nazi Germany experiments. They're kind of torture-y. So scientists don't really like to do it anymore. There's kind of like a negative like stigma around it. Rightly so. So instead, they could use cloned animals to kind of better understand genetics and how genetics work. Because if you clone an animal and then you have like 2 clones of the original, you can do a lot of genetic testing on those 2 clones. Cloning can also be used to obtain desired traits in plants and animals. This is most useful in plants because we can use cloning to make desired traits in like crops and stuff like that. And the last one, which is most interesting, is something called de-extinction. And it's this idea that you can bring extinct species back to life by cloning them. So if we can find like a fossil that contains some DNA of like an extinct species, or if there was like one last rhino in the wild and it got shot, you could like take some of its DNA and then you can clone that organism and essentially bring it back to life. This is especially useful in species that have gone extinct recently because of like overexploitation, like hunting or fishing. It's not like the organism couldn't survive if it were brought back. Its habitat still exists, for instance. It's not like something that went extinct a long time ago and the world has changed so much since then. If you brought something that was like, you know, alive within the past decade and it just got like poached to death or something and you brought it back, maybe if you just like put stronger like anti-poaching protections in place or something like that, that animal would be able to survive still. So it would increase biodiversity, which is good for ecosystems. The cons of cloning. The first is that cloning can result in a lot of cell mutations that just have a lot of unforeseen consequences. The biggest one is that the clone health tends to be very poor. Clones tend to develop tumors. They have compromised immune systems. They age much faster and they have shorter lifespans. And the reason for that is that if you take one of my cells and use that to form a clone of me, the clone of me, when they are born, will be like, you know, a newborn baby, that all of its cells are like the cells of a 30-year-old because I'm almost 30. And so by the time that baby is my age, it will have the cells of a 60-year-old. And so because of that, they again age really quick and have very short lifespans. Lack of diversity is also a problem with cloning. The more clones you create, the less genetic diversity there is. And as we learned about in Bio 20, the less genetic diversity you have, the less adaptable that species becomes, the more vulnerable it is to disease, changing environments, and in the end, extinction. And then de-extinction. We talked about this as a pro of cloning. It is also a con of cloning. Some species that are brought back from past extinction, especially if it is a species that has long been extinct, can bring back diseases that had also gone extinct with that species. You might also bring back one of these long extinct organisms, and it has some negative impacts on the ecosystem that you put it into, because that ecosystem has changed a lot since that organism was alive. So maybe there are new species in that ecosystem that didn't used to be there. Now when we bring this extinct species back, it like kills off all of the species that are living there right now or something. So you just don't always know what the effects of introducing a long extinct organism will be. With that being said, there definitely are scientists that are currently looking into bringing back the woolly mammoth. So, you know, it might happen in our lifetime. And then the last one is that some people think that we shouldn't clone because it's not natural and would not respect the individuality of life, and that you might be trying to play God when you clone things. But yeah, the biggest reason that it is illegal to clone humans is because of the very poor clone health. And then the last topic that we're going to be going over today is stem cells. As we've talked about before, stem cells are unspecialized cells that can be made to differentiate into any specialized type of cell if you just stimulate it properly. Stem cells can undergo much more cell divisions than any other normal healthy cell. Zygotes and early embryos are made entirely out of stem cells, and by early embryos I mean like a blastocyst and like maybe like the first week of life, and that's it. Adult humans have very few stem cells, found mostly in the bone marrow. Some plants and animals will retain stem cells throughout their adult life. These are usually animals that can like regrow lost body parts, like starfish and stuff. And then umbilical cords and umbilical cord blood also contain a lot of stem cells. These are the pros of stem cell research. Essentially, stem cells have the potential to treat a ton of different diseases and injuries. They're all kind of on this list here, but everything from stroke to Alzheimer's to Parkinson's to traumatic brain injuries to needing a bone marrow transplant to spinal cord injuries to arthritis to Crohn's disease to cancer, diabetes, muscular dystrophy, heart attacks, blindness, deafness, baldness, anything you could think of can be used to, the stem cells could potentially be used to treat those. If a person has cells that are not functioning properly, stem cells could be used to replace them. So even if you think of like MS, which is a horrible disease in which the myelin sheaths of your neuron degenerates over time. Well, myelin sheaths are a type of cells. They're made of Schwann cells. So you could take some stem cells and just convert them or differentiate them into Schwann cells and use that to treat MS. So there's tons and tons of different diseases this could treat. Also, like cloning, stem cells can also be used to help with the organ shortage. If someone needs a new organ and they can't get it off the transplant list because there's just not enough out there, stem cells could be used to grow an entirely new organ. And this is something that scientists are currently working on, is growing organs using stem cells. It's very, very cool. They can create like, they call them like ghost organs because they tend to be white before they fully differentiate. But yeah, they're really cool. The cons of stem cell research is usually the controversy specifically around embryonic stem cell research. So the easiest way to get lots of stem cells is through embryonic stem cells. Most women nowadays, not most I should say, but lots of women nowadays like to keep the umbilical cord stem cells because that could be used to treat their own child in the future potentially. And while even if that's not the case, umbilical cords just don't contain enough stem cells, and you just have to keep waiting for like women to give birth and then give you the umbilical cord blood. So it's not a lot that you get out of that, and obviously harvesting bone marrow from adults, not very useful, incredibly painful. So the easiest way to get stem cells is through embryonic stem cells. However, when you get embryonic stem cells, what you're really doing is taking a blastocyst and removing the inner cell mass. And then the inner cell mass is made up entirely of stem cells, and those stem cells can then be used as part of your stem cell research. So that's kind of what this diagram here is showing. You have your morula, it differentiates or develops into a blastocyst, and then you remove the inner cell mass, put it in the petri dish, and then depending on what kind of stimulus you expose the inner cell mass to, it can differentiate into any type of cell that you would want to differentiate it into. All of this, by the way, would be happening outside of the body. Like even the formation of the morula, the formation of the zygote before that, all of this would be happening outside of the body. But the church and many other people do view this as destruction of human life. So this is strongly condemned by a lot of different religious groups, including the Catholic Church, who says that this is essentially murder. That if you form a zygote, if you fertilize an egg in any way, that is now a living human. And so if you then take the blastocyst apart so that you can use the inner cell mass for stem cell research, you are killing that human that is developing there. So there was a lot of controversy around that. I know when I was in school, the Terry Fox run was like a huge deal, and we used to raise like, you know, tens of thousands of dollars every year, my school did, for the Terry Fox run. And I had one teacher, Dr. Graham, who was my religion teacher, who very strongly opposed raising money for the Terry Fox Foundation, because while the Terry Fox Foundation supports lots of different cancer research, they did also support stem cell research, at least back then. So he was very much against that. So yeah, just that's kind of the big reason that stem cell research hasn't taken off, is because it's opposed quite strongly by a lot of religious groups. Okay, and that's it. Let me know if you have any questions.